Cool. Yeah, I think if you start now, that should be fine.
Okay. Yeah, so welcome. My name is Christoph Weber. I'm running a group for the MPI of Complex Systems in Dresden. And the theme of my talk today is I hope I can convince you that the physics of phase separation is very relevant for living systems, not just for modern living systems, but also for systems at the origin of life. Let me maybe quickly just give you a broad introduction to the field of liquid-like organelles in living cells. I have to say that this is a biased perspective on the field because I'm a theorist. So if you find this kind of field and the research interesting, I would highly recommend also to watch some talks from experimentalists because yeah, they are looking at the cells on an everyday basis. I look at the equations for the cells on an everyday basis, a different perspective. So my talk will be much more focused on the physics and how the physics can help to understand phenomena in cell biology. But let's get started with living cells. And come on. Okay, fine. I'll use this one. So if you think about a simple sketch of a cell, you could think about this simplistic sketch of a epithelial cell or any type of cell type. But usually surrounded, it's like a bag, so it's surrounded by a membrane, which is made of proteins and lipids. And it's full of what is called organelles. And these organelles typically have a membrane, also again built of membrane of lipids and proteins. A nice example are mitochondria, and you might know what mitochondria are good for. They're actually kind of the powerhouses of the cell. They produce the corresponding fuel, so the ATP that is then used in downstream chemical reactions. There are other membrane-bound organelles like the nucleus, which hosts, for example, the genetic material of the cell. Nevertheless, there are many other type of organelles that do not have a membrane. They're called membrane-less organelles or biomolecular condensates. And actually, because the community is kind of exploding, they also make up all the time new names for these kind of guys. From a more physics perspective, and I think that's also the most exciting aspect, they share all properties or physical properties of liquid-like droplets. So it's a bit like they behave similar to oil drops in water. So they're typically round. Once you bring them together, they fuse. They have a well-defined viscosity. They have a surface tension. They also undergo coarsening. It's called also oswald-ripening. We'll mention this later in more detail. And so they share all these properties of these physical drops. And here I just cherry pick a few examples. For example, drops inside the nucleus, so really tiny drops. And this is here, for example, the C. elegans embryo, which is about to divide. So it's like the first kind of fertilized egg. And you see before the cell divides into daughter cells, you have these kind of little drops here labeled by GFP, and they're called the P granules. And there are many other examples of the Kadra bodies or the P bodies. And I mean, I really have to say, because the field is so big at the very moment, I guess, maybe the number of covered paper per week is larger, but maybe not too much, because the field is actually going to, there's a review published every 2 weeks in that field. So I just cherry picked here one, which I prefer a lot. If you want to have more references, so don't expect the references are complete, otherwise I have to give like hundreds of references in the field. But that's a very nice review article if you want to have a look what is the number, what is the variety across different cell types of these kind of membrane less organelles. And here I have a simple movie of a protein drop that actually kind of depicts in a little simple movie what actually is actually happening. So you have 3 drops nearby, and you see them fusing and then relaxing to a spherical shape, as you would expect from a condensate that has a surface tension. The key question in the field is not to look for more condensates with liquid-like physical, with the behavior of liquid-like drops. The goal is to understand what they are good for. So what is the cell actually, is the cell benefiting from these condensates? And these insights are actually pretty recent. So this was a sketch in a review article 2 years ago. And actually the papers I'm citing here, for example, are rather recent. So it's like the last years, and it's again, I make a dot. It's just really my biased view. I just add a few references. just to pick a few examples. Let me give you a few examples what droplets are doing in living cells. So one big kind of interest is related to the control of the formation of polymers or fibrils. So it can be pathogenic fibrils like amyloids, or can be also like really functional kind of biological fibrils like actin or microtubules. And people have shown that actually that these kind of condensates can kind of accumulate the corresponding monomers that are prone to aggregation. They can form these fibrils. And then the existence of the compartment, particularly its physical properties, can kind of control the average length and also the average number density, also distribution, by the way, but of these fibrils. So having drops or not having drops actually can act as a kind of a controller for this activation kinetics. Another nice example is that these compartments can make cells more robust against stress and thus deliver more fitness to the cell. So a cell that has certain proteins that can actually create certain compartments. So actually, there's a very famous compartment called stress granule. I mean, because it really has stress inside that once they form because of the response of stress, and now they have shown that they actually kind of protect important protein machinery that is super expensive. So if the cell loses this kind of protein machinery, the cell kind of die because it's kind of it's too much effort to kind of rebuild all this machinery. Another kind of basic idea in the field is that they act as intermediates, kind of that you see compartments forming, in particular at cell membranes, and then they recruit downstream further kind of molecules, and then this entire thing is maybe no molecular drop. It might harden and create kind of more solidified assemblies in the cell. And a nice example are, maybe you've heard of it, like the tight junction or the adherence junction that kind of connects two cell boundaries and thus creates in the end kind of well-connected and also dense or sealed tissue. And a very recent, beautiful idea was is the effect of buffering and of fluctuations. And we're not explaining enough of fluctuations, but very quickly for at least for the average concentration. Think about the same cell type but different cells. So you have a petri dish, we have different cell types. These cells are not identical, even though they have genetically the same. So they might have a different number of factories creating the proteins called the ribosomes, or they might slightly have different volumes, etc. Thus, if you look at specific proteins, the average level of the proteins differ from cell to cell, even within the same type of population. Now imagine this protein has to kind of is involved in some chemical reaction. You know that, of course, concentration levels are key for chemical reaction rates. You want to maintain a certain rate. You don't want to avoid that it happens too fast or too slow. Phase separation can help you a lot, because phase separation actually then phase separates into two compartments, and the difference of the average concentration actually only shows off in it's kind of buffered by compartments of different size, but the concentration levels remain the same. So that's a beautiful effect. So a really fundamental aspect here, I think it includes in both this aspect here, also the control of aggregates, is in general the control of biochemical reactions of these kind of compartments. And in my today's talk, I thought about like giving you first of all a broad introduction to the field of droplets in cells, but then also cherry pick kind of two research kind of directions to tell you a little bit more what people actually do in the field, what are the interesting research questions, also give you a few results. And I picked exactly that kind of how well droplets can control biochemical reactions as one theme, because it's so relevant for the cell. Okay, you should not picture, of course, this kind of physics of phase separation in the cell as, it's like having oil drops just in the cell. It's of course not true, because it's a living organism. The cell relies on a continuous influx and outflux of energy. I mean, that's kind of a fundamental property of life. That's kind of what makes it different to that matter. Thus, also these condensates live in an environment, in a non-equilibrium environment, and are generally affected by this non-equilibrium environment. And thus, we have recently introduced new kind of theoretical concept in the field that we call chemically active emulsion, to make a difference to classical emulsions, so meaning droplets composed of many drops. And that's why I devote now 2 slides to this kind of theoretical concept of a chemically active emulsion. But I do it step by step, and please interrupt me if you have never heard of what I'm talking about. So I will do it step by step, and assume that you have not seen maybe the theory of phase separation of a classical emulsion. If you, of course, know that, please let me know. So let me start with a classical emulsion first, to then work out step by step what we mean by a chemically active emulsion. So emulsion is simply a system composed of many drops, okay? And they undergo kind of what is called ripening in the case of drops, or in general in physics, coarsening. So imagine you have 2 drops, different size. Once they come together, they fuse, and in general, there's also Oswald ripening. Oswald ripening is one pathway where in the end the smaller drop shrinks, simply, and it relies on the fact that droplet interfaces have always a surface tension, and there's a curvature of the boundary. And this affects the concentration levels right outside of the drop, actually such that you get diffusive fluxes from the small to the big one until, so this will then shrink, this will grow, until this will, the small drop will eventually disappear. So how would you now describe this from a theoretical perspective? You would write down simply a kinetic equation that tells you about how the concentration field evolves in time and space. And I will discuss here the most simple kind of basic model, and this is the one of a binary mixture, meaning a mixture composed of 2 components that is incompressible. So I've only worry about one concentration or one fraction field in the system. I neglect here flow for a second just for simplicity, just to get the concept across. And so the time changes of this concentration field actually are then governed by a conservation law, which you see here by the divergence of this object here, and this object is exactly minus the flux. And the flux, or the movement of molecules in a mixture, is generally driven by gradients of chemical potentials. It's very important, it's not the gradient of concentrations, because this is only true for dilute systems. And here we have a system that needs large enough concentrations in order to phase separate, so it's really driven by gradients of chemical potential. And mu bar here is the chemical potential, and this capital omega is here is the mobility. So how is this chemical potential now linked to thermodynamics? Is simply linked by a derivative. To be slightly more picky, it's linked because by a function derivative with the free energy, because the free energy is actually a spatial integral of a free energy density. So just like that includes the interaction, that includes the entropies or stuff that you know from your thermodynamic lecture, but also it includes a penalty to create gradients. And when you think about coexisting phases, there's always an interface in between, and it costs energy to create these kind of gradients. Thus, it's kind of intuitively clear that this penalty parameter that kind of tells you how much free energy you have to pay to create a certain gradient is kind of linked also to the surface tension, because the surface tension tells you like how much energy is in an interface. So what is this system doing at equilibrium? I mean, I think you have seen this in your thermodynamic lectures. So in order to get phase separation, it's pretty much the same idea as for liquid gas phase transitions. You need to have a free energy density of this kind of shape, okay? It's kind of double type of shape. And thermodynamic equilibrium corresponds to the case where the chemical potential is homogeneous in space everywhere. And this amounts in the end to looking for the concentrations that have the same slope of this free energy density. Because the chemical potential, as you can see here, is the slope of the free energy. And that tells you that, okay, you get 2 coexisting phases at equilibrium. That's exactly classical phase separation. But in a finite size system, meaning realistic systems contain finite size drops in a finite size box, these concentrations are actually slightly upshifted because of surface tension and the fact that, and the size. But this actually, once the system gets larger and larger, you perform this classical thermodynamic limit. In the end, kind of is irrelevant, and this blue dot kind of merges here with this orange dot. Okay, now let's add chemical reaction. What changes in this very simple system of a binary mixture once you add chemical reactions? And the most simple case you would like to study first is a simple chemical reaction between A and B. So A becomes B and B can become A. So how would this kinetic equation change? It would change as follows. I mean, you have still the fluxes that move the molecules in your mixture, but you would supplement this equation with a chemical flux. But this chemical flux for a system that that approaches thermodynamic equilibrium has a very important constraint. So this chemical flux has to vanish, as the chemical potential is 0. Huh. What does the chemical potential 0 correspond to? It actually corresponds to the global minimum of this free energy. Because now the system can actually walk along this line. The coexisting system, the phase separation couldn't do that. It had to conserve the total number in the system, so it could only split up into two phases of different concentrations. Now the system can walk, break kind of conservation of mass, and then go to the global minimum of the system. So a system that can phase separate and have this simple chemical reaction will actually go to a boring, homogeneous state that corresponds to a concentration sitting at this minimum of this free energy density. But that's bad, because now it came up, okay, cool, that's phase separation, now I add a chemical reaction, it's gone. So what's not exactly the point? Now it's homogeneous again, it's kind of just like an extra term you have to deal with. The chemically active emulsion actually differs because it drives, it makes sure that this chemical reaction cannot equilibrate. So it continuously uses some energy to push this away from equilibrium. Let me explain this step by step. So for that, we have to rewrite this chemical flux of the system a little bit. First of all, the thermodynamic system. And let's do it. So let's just do the thermodynamic system. So how would you do it? But there's a very important relationship for a system that can relax to thermodynamic equilibrium. So the forward and the backward flux of this chemical reaction are always slaved, okay? So you have a relationship that's called theta balance with rate relationship between the forward and the backward flux, and it's exactly slaved here by this Boltzmann factor. And you see that once I'm at equilibrium, so this chem potential is 0, the exponential of 0 is 1, you exactly are at equilibrium, so the forward and the backward flux is the same, so the total reaction flux is 0. Beautiful, equilibrium. So what is now a chemically active emulsion? We just add, okay, then you can, you can just rewrite this total flux into a forward and the backward flux, and what you see then is that there's a single rate constant that tells you how you fast approach chemical equilibrium, and a term here in the bracket that actually sets the equilibrium, okay? So because the rates were not independent, only one rate. And this differs when once you have a system that is continuously driven. Then we usually say theta balance rate is broken. So let me illustrate it by like an academic kind of way of illustrating it, and later tell you how biology is actually doing it. So in addition to this kind of thermodynamic pathway here, I add another one with a where I introduce here a fuel and a waste, right? And first of all, this is reversible, so it's also looks like a thermodynamic chemical reaction. So I can just write down two relationships, two theta balance of the rate relationships of this kind. So for the first pathway, 1, right? And for the second pathway. And the only difference here with the second pathway is I have a new chemical potential here, which is simply the difference between the fuel and the waste, okay? And now let's do the following. We can do a little trick, of course. We can just write the forward pathways, just the forward fluxes in terms of two pathways, the backward flux with the two pathways, etc. And then just calculate the fraction. Then you actually just get this, but this little algebra trick, you just get this beautiful expression. Now imagine the following. Imagine at, for example, the boundaries, we maintain the chemical potential between the fuel and the waste, which corresponds to just like always supplementing fuel in the system and removing waste, okay? So we drive the system away from equilibrium. So from the perspective of just looking at A and B, it looks like the theta balance of rate is broken, okay? And you see it very nicely here. So once this value is non-zero, you see that I get an independent forward and the backward flux. If this is 0, for example, then I get right over here 0, and then you are back to the normal first pathway here, okay? And that's, of course, a rather, let's say, academic way of saying breaking theta balance of the rate. I mean, how is biology doing it? I mean, I've shown you, okay, fine, you can, by the boundaries, drive the system away from equilibrium by just maintaining the chemical potential difference between fuel and waste. But biological systems do it slightly different. Does do it usually in a much more harsh way. They have often irreversible reactions. So in the end, the fuel is, so to say, burned in this reaction. You produce a waste, but in an irreversible manner. And that's how typically many biological systems actually break theta balance of rate, because they are, practically speaking, okay, for the super experts, there's always a little back arrow, but it's often that tiny that it only matters maybe on universe timescales. Okay, so this is the idea of a chemically active emulsion. So it's continuously driven away from equilibrium, and thus it can interact with the physical phase separation. So remember, before I told you like physical phase separation, we had this chemical reaction that can react with thermal equilibrium, no drops. Now I break theta balance of rate. I hope still droplets can survive. Yes, they do. And I just show you a few movies to relax a little bit from the theory here, what you can actually see. So already a few years ago, a very beautiful paper from David Zwicker, a friend of mine, he has observed that you can suppress ripening. You can suppress, in particular, oswald ripening once you degrade material inside the drop, and produce it outside. And then you get, in the end, kind of a monodispersed emulsion of equally sized drops that even form this very beautiful hexagonal lattice. But you get even much more. You get even like very funny rings that are a little bit reminiscent. That's also called like patterns in active emulsions. To I allude a little bit to reaction diffusion systems, Turing patterns, and this kind of stuff. You can also see the formation of rings, and then they show very funny instabilities, and they can break up into these little drops. And recently, we have also seen, for example, that these rings can even be stable. We have not seen so far propagating rings, but it's just, I guess, a question of time until, or a little bit of a matter of crazy fueling to see also propagating rings, I think. You should really note the difference to a classical Turing system is that the system, if I switch off the fuel, we are back to the thermal equilibrium system. So we have phase-separated drops that do what phase-separated drops do. They undergo co-stirring, okay? So once you add the fuel, of course, somehow you loosen these thermal constraints, but still you have well-defined drops, so with the coexisting phases. But of course, if you really drive the system very hard, at some point you might end up into a system that looks mathematically and also at some point physically like a Turing system where you only have diffusivities and the pattern is purely driven by the chemical, by at least quadratic biochemical chemical reactions. You can also go to 3D. I mean, you can look at these patterns in 3D, but you can see also another phenomena that we recently published, and this is the division of drops. So you see, that's a phenomenon that you will not get. I will play it again because I like the movie a lot. This is a phenomenon you would never see, and it's a forward movie, okay, I have to say that. You will never see an equilibrium system because there, of course, you bring drops together, they fuse because the resulting drop has, or the resulting system has a lower surface, interfacial surface, and thus has a lower surface energy. And thus the system cosines in order to minimize the free energy, in particular, the free energy kind of hosted in the interface. And here they do the opposite. They create more interface, they divide and divide, but this exactly comes from the fact that the chemical reaction pushes the system away from equilibrium and then can fight against surface tension. So if you ramp up the surface tension, this will not happen. So then still the droplet always, you perturb it, it will always come back. If you wonder, like, okay, what's the underlying detailed mechanism, how that works, I can a little bit make it a little bit quick and give you at least an intuition how that works. Once you have a droplet and perturb it a little bit, there is like a well-known instability. It's called Moulin-Sickerka instability that people have already found for solids in the past. This just tells you that you typically, once you perturb it, have a tendency to grow at the large, it's hard to see, here, at the large curvature edges. So the droplets, or this kind of Moulin-Sickerka instability tells you that it kind of would grow and kind of maintain at some point the aspect ratio, keep on growing, but it stays deformed. It doesn't go to a liquid like that. But here's a difference. I mean, it has some similarities to this Moulin-Sickerka instability, so you perturb it a little bit. It wants to grow in that direction, but then there's a huge difference. We only see division in the parameter space where the droplet roughly maintains its total volume. So what can you do once I tell you you have to elongate along the small curvature edges, so you go like this, okay? And you have to conserve the total volume. You can only do it by creating a neck, split it into two. So for the people who are a little bit more interested, I can give you like also an analogy then to the electrostatics, but we can also do it at the end of the talk. Okay, so why should we care about these nice theory movies? I told you already, okay, droplets are relevant for cells. Once they're driven away from equilibrium, we haven't seen these dividing drops in living cells so far, but we have seen that they organize biochemistry, and we'll talk about this a little bit later. But I think that this movie is in particular interesting from another field, and this is the field that asks questions about the origin of life. So if you wonder what is relevant for some early life, I'm talking about really like simple life, a few molecules, and they have some cool features. You would always, I think the field has also agreed on one fundamental aspect, that in the end you need compartmentalization. So you need in the end a distinct identity of an object you talk about that is different to the outside. That's kind of a compartment. Whether it's a membrane-bound kind of object or liquid-like drop, of course, that's a matter of viewpoints maybe, because we were not present, obviously, at the origin of life. But there are, of course, others, because, of course, for example, droplets offer you a distinct chemical environment inside. So that's a little bit the basic idea of a microreactor. So the inside is different, the reaction rates are different, I can accumulate molecules, and that's ideal kind of to run biochemical reactions inside. But also you can host chemical reactions just inside, which do not exist at the outside. And what life definitely needs, a kind of a fundamental property, is kind of a metabolism. And if you would kind of really reduce it, I mean, the metabolism in humans is very complicated, but it's just some basic key features. And this is, there's an uptake, I think that all living systems share this fundamental property. There's an uptake of nutrients that you convert into fuel that in the end you use in some other kind of downstream process to regulate some function within kind of this kind of living organism. In our case, it's typically the fuel. When I talk about fuel, I mean ATP. But it always comes with waste production, right? So the entire point of life is to consume nutrients and to create waste. That's kind of fundamental property. And that's, of course, what droplets can do. They can host these kind of biochemical reactions, exactly suck in material because they're liquid-like pretty easily. And then, of course, this movie I've just shown you is kind of ideal. So once you have this, yeah, that's kind of something that some people already call kind of pre-biotic life. But then imagine, then you want to make more of yourself and then go to this kind of population genetic kind of fight with other kind of compartments that are different, that has different metabolism. But for that, you have to get more, create more out of yourself. And that's, of course, then this division idea here is ideal kind of to deliver this for free. It's just physics and chemical reactions driven by equilibrium. Then there's another kind of question in this field that's also associated with phase separation, but I glimpse over it very quickly and come back to it at the end of my talk. There's a big conundrum in the field, and it's related to this very early origin of life, where in the end the Earth has kind of relaxed a little bit, and you have only a primordial kind of mixture composed of DNA, RNA, and proteins, so small chain kind of peptides. And these are all, when you see this from a physical perspective, these are all kind of polymers. But the problem is the sequence space of polymers is huge. Thus, a specific sequence is extremely diluted. If something is diluted, you can't do much with it, right? I mean, it's diluted, it just hangs around, there are no chemical reactions, no phases, there's nothing interesting can happen. So there's a fundamental conundrum. How do you select specific sequences out of this humongous high-dimensional pool in order to just only use a few? With a few, I mean, if you look at modern cells, they have 10,000 different components. It sounds a lot, but it's, of course, tiny compared to the exponential sequence space when you just build a long chain DNA, for example. And also to how to enrich molecules in order to make sure that they really can run biochemical reactions. And then I will show you later on a concept actually how to solve this. And actually one can do it with phase separation, which is pretty cool. Kind of to move a dilute pool of a lot of polymers into a very dense pool where you have only selected a few specific sequences. Okay, I'll come back to this later on. Okay, this was a little bit the overview of the field. I mean, what compartments are good for. A quick summary: intracellular chemistry is very important, living cells, but also the origin of life field is very excited at the very moment about these ideas, kind of to use the physics of phase separation to, I would say it's not the only solution in the field, of course, just to provide an additional mechanism that could have been relevant at the origin of life. Let me say it defensively. Okay. So now in the second part, I would like to give you a glimpse of what we do from a more science perspective. So this will be switched slowly more to a science talk. And I cherry-picked two topics also in my group that are associated to what I just mentioned already. And by the way, interrupt me anytime. Feel free to ask. And I picked exactly topics from these two different fields, one from intercellular organization. So what we do actually, we look at different type of chemical reactions, so very simple kind of physics-minded, simple chemical reactions. For example, like nucleation, like the formation of aggregates from monomers, for example, where you need fuel. That's kind of the biochemistry. So we always spend the energy to provide the chemistry, to provide a chemical pathway. Or, and that's the example I will focus on today, to activate molecules. So kind of a two-state model, right? Where you have kind of a molecule can open or close, has kind of two states. It's super important for many little folding transitions, where you use fuel to activate the components. And then we know how to describe this for a homogeneous system. And then we just ask the question, once you have drops, how does this change actually? As a spoiler, it can dramatically affect it's the biochemistry. And then, of course, in my group, we look at, of course, not just these simple kind of cases. We also look at protein expression, enzymatic reactions, and more complex reaction cycles. But this gets then in more technical, just more parameters, but the same basic idea. And then the second, the last part, but it depends on your question and also the time that is left, I will kind of give you this glimpse of how cyclic phase separation can kind of solve this conundrum of having very diluted and sequences in primordial soups. Okay, then let me get started. So I cherry-pick this kind of example of this. You have a deactivated component, you want to activate it, you use fuel for the forward reaction, and kind of the backward reaction is kind of spontaneous. And that's a system that you look at. So it's some kind of a finite size container that, of course, should kind of represent the cell, so to say. And inside there's a drop. Of course, the drop is typically much, much smaller, but just because of illustrative purposes, it's much bigger here. There are two, the coexisting phases I denote as 1 and 2. So the red stuff is kind of the droplet material, or that's kind of the droplet that is made of droplet material and has a certain volume V1, V2, and that's the entire system has a certain system size. So how to describe this system? Actually, the machinery is very simple. It's very similar to what I already have shown you at the beginning of the talk. So you write on these kind of kinetic equations, but the only problem here is you have just more components. So the minimum model for this chemical reaction is you have a solvent, you have a droplet material that makes the compartment, you have the A material, the D material, and the fuel. So if you have at least a five component system with a lot of parameters, but that's how we usually get started with. So we write down in the end the full set of equations, break it depends on the rate, similar to what I did. The only difference is we have more parameters and the equation is simply more messy and a little bit bigger, but it's the same kind of basic concepts. Today I will would like to show you, get you close to the glimpse of what how the biochemical reactions are kind of affected. And thus I will use kind of two assumptions that will simplify this complicated theory dramatically. And first of all, I would like to consider all the reactants, so the activated, deactivated component, and the fuel as being dilute. Okay, and this leads to a very simple picture. So meaning what actually happens if they are dilute, they have zero volume. So the entire compartment that is made of this droplet material in the end just sits there, and the AD material and the fuel just diffuses in and out, but doesn't create growth or any coupling. So it doesn't affect the phase separation, but these reactants see a difference between a homogeneous system and a system with drops. And then the equation that you obtain, I mean, it's rather intuitive, are in the end effectively reaction diffusion equations. They're non-linear, but that's a detail, in a field of this kind of droplet material. So this is a typical profile of the droplet material. So if you would start here at the center and would deviate here radially, and then here's the interface, you would get here this drop down in terms of concentration. You go from phase 1 and phase 2, and that's the droplet radius. So just walked here. And that's the type of equation we can solve. We can analyze pretty easily. There are standard methods to look at moving like stationary or transient coupled nonlinear diffusion equations. But for today, I will simplify this a little bit further. So that's then a stronger restriction on a certain kind of subset of activation, deactivation reactions. But it's a very simple limit. And this limit is the limit where the diffusion is very fast, so the molecules diffuse quickly, and the chemical reactions are slow. And then we end up with a set of ODEs, so simple ordinary differential equations, just for the concentration of A and D inside and A and D outside here. So we have just then 4 fields. The only hidden further assumption, I come back to this in a second, I just maintain the fuel constant in the system, so I always keep on fueling the system. So kind of that's basic logic. So I don't want to let the system starve or play with the fact that I can overfuel it, or I can starve it. I mean, we do this with experimental. It's pretty funny. Once they starve, they show also very nice physics. But I don't do this here today. But we have just these 4 fields here then. I have to be slightly more precise here because I cannot just say fast diffusion, slow chemical reaction. It sounds very unphysics, right? Because like fast compared to what? So actually what we have to compare are length scales. And the length scale here is exactly the length scale you would find in a reaction diffusion system. So that's here the diffusion constant. And this is kind of the first order reaction rate. So it's kind of this forward and this backward rate, right? And for this equation to be valid, actually, you have to make sure that the system size is smaller than this kind of next scale here. And then what I just showed, if this stuff is rather dilute and this is obeyed, the equation I write down are absolutely correct, okay? But they are much simpler than those equations, you can trust me. And so what is then exactly this drop material doing to the system? It introduced the notion of partitioning. So it introduces 3 parameters for, in our case, so for D, A, and F, also for the deactivated material, the activated material, and the fuel, you get something like so-called a partitioning coefficient, which is it's a very simple, very important object in the field, but it's a very simple one. It's just the concentration inside over outside, okay? And these are the key parameters that we'll actually then exploit and play a little bit, and next to the compartment volume. As I said, I maintain the fuel level constant. This is kind of, so we just have a pipe, we just add the fuel under the system stuff, and then you actually end up with 4 set of ODEs. So here's just the first 2 ones for the deactivated material inside and outside, and they have a very intuitive structure. They have this classical mass action structure that you would have guessed right away. This comes from the dilute limit, where you have, for example, here just like the forward pathway, where you out of an sorry, that's a backward pathway, sort of you make out of an activated, a deactivated one. It scales exactly without concentration, and also the opposite pathway. That the only difference here now, it requires now fuel to move from D to A. But there's a very intuitive structure, and there's similar kind of equations with a minus sign, because the total A and D is kind of conserved in the system. You would also have here then for the A material. So what's now the essential interesting new term? That's exactly this one here, those guys here, okay? What do? You see here these brackets, right? They try to push they create a flux until the flux is only 0 once the concentrations inside and outside are identical to this partitioning coefficient. That's the case. If it would switch off the chemical reaction, then these reactions would just equilibrate according to the partitioning coefficient. And this comes, of course, with a time scale. And once you do the derivation, you can get this time scale, of course, explicitly in terms of the more microscopic theory, and you will scale with the diffusion constant, droplet size, and these kind of aspects. But this will kind of can be compared then, for example, to the forward and the backward rate. So there's a nice competition of time scales, how fast you equilibrate the partitioning versus how fast your chemical reaction kind of changes the levels. Okay, so what do we get? First, we have to ask a question. Now we can solve those equations. Actually, pretty simple, easy to solve. We can even get analytic results for this. We have to ask a question, and the simplest question I ask now here is, how does, let's say, we just look at the activated component because it sounds more fancier than the deactivated one. How does the average concentration for large timescales, I look at a stationary state, okay, with drops compared to the concentration without drops? And I just define a simple quantity, okay? If this kind of quantity is kind of larger than 1, then I have more activated components compared to the case without drops, and otherwise I have less, okay? So let's have a look, and we get, for example, these kind of graphs. Let me guide you through step by step. So I just plot here this quantity here as a function of compartment volume. Here at 1, that's kind of our reference system. That's kind of the homogeneous system. So once all the curves are above, I have in the end more activated components. Once they're below, I have less. Okay. So what is now different between these bluish curves and the red curve? The essential difference is, this is also shown here, is in terms of the partitioning coefficient. For the blue curve, the deactivated component and the fuel partition both into the drop, okay? While, for example, for the red case, they partition into the opposite kind of phases, okay? So we see that we get actually already a kind of amplification of the average, the amount of activated components once we kind of bring the components together in the drop, and we get the opposite. So we see overall that we have a control of the chemical reactions. First of all, we should also recognize, okay, cool, by playing with the volume and the partitioning, I can dramatically affect this. And here it's a log scale. Have a look. I mean, it's not like a factor of 2 effect. What's the underlying mechanism? I mean, you should also appreciate here, I've plotted here two blue curves. So this curve actually has a smaller partition coefficient. So as I go from this light blue to this dark blue one, I just increase the partition. So the kind of the tendency to go inside the compartment. And so what is the super simple picture for the mechanism, or what's the underlying mechanism? The simplest idea of a mechanism is simply that, of course, in order to get more activated components, of course, we have to kind of co-localize the fuel and the deactivated component, because they make, in the end, the activated component, okay? But it's not that simple, and I'll tell you why. Because then you would prefer to have a very tiny compartment, even a tiny one. And why is this? Because actually, if you want to just increase both inside the compartment, the fuel level and the level of deactivated components, you would better prefer a super tiny compartment. Why is this? Because then in the remaining system, the concentration is the average concentration, and inside it's exactly enriched by the partitioning times the average concentration. But that's strange, because if you would collapse to volume 0, then you cannot have any effect because you have no drops, right? There must be some competing mechanism that kind of tells you that there's a kind of a distinct optimum here. And in particular, you should also recognize this value doesn't not just move up, it also moves here to the left, actually to smaller compartments. It tells you the higher the partitioning, the optimal value, the optimal kind of activation values are at smaller compartments. Okay. So what's this competing mechanism? So I just mentioned this colocalization, anti-colocalization idea, but this is not just it, you need a competing mechanism. So I've just mentioned, yeah, you would just go to smaller drops. So if you would like to move this to the left, to just enhance the concentration of D and F. However, there's a competing mechanism. This just tells you, if I have smaller and smaller volume, of course I reduce my turnover rate, right? So if I have a super tiny compartment, then of course I produce less molecules per time, because the reaction only happens in the end inside the compartment. That's kind of the competing mechanism that kind of pushes you kind of to some optimal value. That explains that if I increase the partitioning coefficient, that also this optimal value goes to smaller compartments. Okay, then of course you can quantify this more. I just give you a few glimpses of a few more results. So for us, it's very important that the values are relevant for biological systems. So if you plug in numbers that you can measure in experiments, is this effect significant? Is it a 1.2 correction to the homogeneous system, or is it a significant effect? And actually, it's a significant effect. So if this is a typical range of partitioning factors that we know for biological systems. So typical partitioning factors go from 1 to 1,000. So much larger values are very hard to get. So I guess in the end, we have to show that in this typical range of parameters, do we get in the end, is this quantity, is it 1.5 or is it a few hundreds? And actually what you can see here in the color bar, it's in the order of a few hundreds, meaning the effect is dramatic. So meaning if you compare a homogeneous system with a system with drops, you can actually, out of the blue, enhance the pool of activated components by 100-fold. I mean, that is, I think, a cool feature biological systems can use in order to control the level of certain components. But for the people who are interested in kind of more analytic approaches, you can run classical machinery. So here, that's another plot. That's kind of how the optimal volume changes. I mentioned, like, if you increase the partitioning, you go to smaller and smaller optimal volumes, and you can actually calculate this. So you can really calculate analytic expressions, how the optimal volume depends now on these partitioning factors. And just like, for example, when you walk along this diagonal line, you can then verify how beautiful there's a scaling relationship that you can even calculate, including the offset. So these, you see, like, we start with a very complicated system, and which is hard to calculate, but you can boil it down with some reasonable assumptions that are met by biological, that are consistent with biological systems. You can even do analytic predictions, which is, I guess, very nice and almost like good for, at least the theory is almost like it, that they can write, not just solve it in the computer, but also calculate it. Okay, so quick summary. I hope I could convince you just by cherry picking this example. The droplets are great in controlling at least this type of biochemical reaction. I told you a little bit about this mechanism. That's kind of a competition between co-localization and kind of having sufficient turnover. Now we can do the same type of recipe for all kinds of chemical reactions. And this is what we do in my group, for example. And I can tell you right away, and spoiler, the main output actually find the same characteristics for all those biochemical reactions. So droplets, it's really a generic scheme that droplets can affect biochemical reactions kind of dramatically. And now what we do now, we have a lot of collaboration with experimentalists. We really try to show in the end whether cells or living systems have optimized it, or they're just working in a very unoptimized regime. So because as I showed you, there's also kind of an optimal size of the compartment, optimal partitioning conditions. Has the cell evolved to get kind of optimal control of this biochemical reaction? But this we don't know so far, but at least theoretically, there is an optimum. Let's see whether cells actually have optimized it as well. Okay. Wow, I took a little bit longer than I expected. we started, just remind me, we started at 5. So how long should I talk without annoying the people too much? Because it's kind of the second part.
I mean,
Usually,
just go for as long as you want. If we go sort of past like usually questions.
Ask a question. Like, I've just a second story about the origin of life, about how to select in the end these molecules. I can make it slightly quicker, but
I think just go with what you were going to do.
Okay.
I think people,
because
people can come and go if they want.
Okay, cool. Okay, so this is a different story. This is like a story where phase separation could help kind of to select molecules at the origin of life. I've already mentioned that the origin of life I'm talking about is not in this very complicated and also very beautiful type of questions where the life is more complex, you have very kind of pre-biotic organisms already. It's just where in the end chemistry meets polymer physics. So in the end, where you have a soup full of DNA, proteins, and RNA, and you want to create in the end step-by-step more complex structure by making them longer and longer. Okay, and there's a fundamental problem. So currently, people believe actually that kind of RNA, DNA, and say short chain peptides were present in this primordial soup. And actually, there are great chemists. So many conferences that kind of you don't need much actually to produce small chain peptides out of some just in some bioreactor. But there's a fundamental problem, and it's very simple because, like, when you think about the fundamental information carriers of DNA and RNA, we have like 4 nucleotides, proteins that are around 20. And when you just would like assume random polymerization, obviously you get a huge sequence space. Of course, it dramatically exponentially depends on lengths. But so once you are in the length of 50, and proteins and cells have a much longer sequence, you have a huge sequence space that exceeds by so many orders of magnitude the typical number that actually is used by biological systems. So this kind of suggests that living systems might have not tried all combinations, but there have been several successive steps of pre-selecting them, and maybe it definitely involved chemistry or kind of chemical reactions, but maybe also physics, and that's probably the physics of phase separation. And okay, that's a simple point. I mean, I can skip this, that they're rather diluted. It's a very simple argument. I mean, the more sequences I have, it would just assemble polymers step by step, or typically create a new sequence instead of, in the end, creating same sequence again and blowing out, like increasing the concentration. So a really fundamental question in that field is in how to select in this. And so that's a very simple physics kind of picture, of course. You just have a box with lots of so-called heteropolymers. So, I mean, that's kind of the physics language of just, okay, I don't care now whether it's DNA, RNA, or like a protein. I mean, I assume it's unstructured. I mean, that's a different complexity once we talk about structured proteins, of course. But for me, these are just heteropolymers with a huge sequence space. And how is it possible to select specific sequences? And how is it possible then to enrich them in number? Okay. And why is it important to enrich them in number? I mean, very simple, because all the interesting phenomena rely on the actual concentration of these kind of components, because only then I have significant chemical reactions, only then I can think about assembly processes, aggregation, formation of membranes, or even phase transitions. All this requires large enough concentration. So with super dilute kind of suspensions, I can't do much. And people have thought about this in the field a lot. Because of sake of time, I skipped this, but I have to definitely want the credits. People have thought already that the polymerization kinetics can lead to a significant bias in the sequence space. And this is super exciting. And there are different kind of ways of thinking about it, but because of sake of time, and I don't want to make the talk too long, I just skip this a little bit. But I mean, the message simply about the polymerization kinetics can lead to some bias in sequence space, but that hasn't fully resolved the problem. Let me just skip this. But length of a polymer is not always a problem. I mean, I just mentioned it's a problem because you just blow up sequence space, and then of course you have so many possibilities and how to get single kind of molecules out of it. And length is also an advantage. In particular, polymer physicists know this. I mean, that's exactly their main problem, because the longer heteropolymer, the smaller the mixing entropy. So this is range density. And if you have a simple mean field kind of description of a multi-component system, you would write down such a mixing entropy and some kind of interaction kind of among the molecules I and J. And the mixing entropy is known, of course, that it decreases the longer the polymer, meaning as long as you have some adhesive units in your system, even of very low concentration, if they're very long, the mixing entropy goes to 0, meaning as long as they have some weak tendency to like each other, you immediately phase separate. And that's, of course, for us, it's great from in the field of phase separation physics. But for the classical polymer physics, that's something they wanted to avoid. I mean, they want to bring the stuff back in solution, long chain polymers. But here it's really a cool advantage. So you get it usually for free as a cool property. So if you would just guess a system that is composed of adhesive units that makes up a heteropolymer, whether it phase separates or not, I would just always guess, yeah, I mean, there will be some phase separation, just no matter it's like what, but there will be some, because exactly of the fact the mixing entropy is so tiny. Yeah, can you now select with phase separation? I mean, in general, of course not. I mean, there's a different language. You would talk about coexistence of phases. So let's assume the simple system you can think about to talk about kind of selection. There's a ternary system. So you have a solvent, it's incompressible, so solvent is kind of hidden in the simplex, and you have two components, say two types of DNA molecules. And imagine this region here is called kind of the region where you phase separate, and imagine some average concentration, you decompose in the end into these coexisting phases. And when you just look how when you look at the corresponding concentration, so let's say concentration 1, you see like, okay, inside I have de-rich in the end the concentration of 1. Sorry, the concentration, yeah, it's going to have 1, outside of de-rich concentration of 1, and here the concentration of the component 1 has actually enriched here. But you would never say that this is kind of selection because it's just classical phase separation. So you cannot, of course, truly select. But what about cycles of phase separation? So once you think about cycles of phase separation, maybe you can exploit this, but you need some non-equilibrium aspect kind of driver here. And why cycles? Is cycles relevant for origin of life? I mean, cycles are kind of ubiquitous in these kind of systems. And there are two deep beliefs in the field that life has either emerged somewhere deep to these kind of some events and deeply in the ocean, or at some salty beaches and lakes, etc. And why do people believe that? I mean, the common physical properties is that it all happens close to kind of porous structures, and porous structures have amazing properties. Of course, they create kind of extra protection from the surrounding, from what I would call now the pool. They offer surfaces for catalysis, they allow convection, lots of cool physical aspects, but for sake of my talk, they allow in the end also kind of changes. They can kind of isolate themselves because they're kind of seasonal, daily fluctuation of temperature, salt, high, low tide, whatever kind of typical kind of fluctuations in or cycles in our daily life. And then they can kind of open and close and kind of have periods where actually they're isolated with respect to the pool, and sometimes then they can kind of let in molecules from the pool. And that made us thinking of, okay, what about a simple abstract rule that kind of moves? I'm not modeling these kind of systems, of course not. It's super complicated. There are people who tried it. It's really amazing stuff. But we just want to extract the main key feature, and the main key feature is the following. We want to combine phase separation and cycles where you're either exposed to a pool, where you can exchange your outer concentration with the pool, and then you again isolate it for some time period, and then you can exchange it again. And the question is, can you evolve away from the pool by several cycles of phase separation? And the quick spoiler is yes, you can, and I'll show you now how. So now it's an abstract rule. So we have to stand like, now we just take these two principles and test it first of all just with a simple kind of, that's why I call it also now protocol. Okay, it's not a model for a thermal event or anything like this. The protocol goes like this. So I have a box here of all these heteropolymers. We make them phase separate, but just evaporate in the water or anything like this, and DNA usually always does. Then we cut out here in such a dense phase, throw it into a new kind of test tube, and fill it with the pool. And the pool is the reference system we want to evolve away from, right? So kind of, and then what we do, we just heat it up, for example, make it homogeneous, and we look at this kind of composition. This is kind of one step of phase separation, okay? And then we ask ourselves, do we kind of evolve away from this pool that we add? We always add the same type of pool, so we neglect now potential time changes of the pool by several cycles of phase separation. And actually, this actually works. And what we always look at is kind of look at the system after each cycle and see how it evolves. And I depicted on really the basic principle. We do this really for a very high dimensional mixture up to many hundreds of components. And here I depict just the principle for, okay, here first of all the model comes. Okay, I glimpsed you very quickly over it. We discussed already this free energy. interactions that determine some, of course, the phase separation, and we have to think about how we fill in the end this interaction matrix. That's exactly where we establish a pipeline, how to take in a given pool of certain sequences, how to calculate the interaction matrix. This is a very technical aspect, there are different methods in the field, but we have a way how to do that. And then the goal is kind of to create in the end the concentration of the inside phase. So it's a composition vector of all the components. And then we just put this into the cycle update rule of mixing that I've just illustrated. So don't think about the mathematics, it's not that relevant. What do we observe? The first naive feature we see is when you think about it, it's not very spectacular, but it's somewhat pretty cool still. You see, once you go over several cycles, so this is now the cycling time, so we cycle one after the other, we see that the dense phase, the initial dense phase, blows up and becomes the entire system. Which is pretty cool because, like, wow, we had a dense phase and now it makes up the system. So somehow I have, if we imagine we had selected in this pool, right, in this kind of system with respect to the pool, we have definitely solved the conundrum of too low concentrations because the system just became the dense phase. Okay. Do we now select? Yeah, this you can illustrate. I just illustrate here for the simple ternary system. Because of course I cannot illustrate a 100-dimensional mixture here in the diagram. So let's start with the pool. Let's really initialize the system with the pool and then do exactly these mixing events that I've explained. So once we undergo then what we do actually, we walk along the tie line, and these lines are called the tie line, for each mixing event until we crush exactly at the right binodal. That's exactly what you just saw. I mean, that's the growth of the volume. But as you do this, you see this here, all these kind of, so here plot the volume fraction of these two components. It's of course cherry picked. I mean, we just choose some interactions to illustrate the principle. You see that component 1 gets kind of enriched, while component 2 gets kind of enriched, okay? And that's a very simple idea, very simple-minded. If you don't start with the pool, you have some fun introductory that's kind of nonlinear here. Yeah, it's just like this because you can even calculate it analytically. Then you can move this on to multiple components with random interactions, but with our collaborators, actually, we now write down kinetic rules to create pools and also look at truly sequence pools and then predict and compare this with experiments at the very moment. But here we just scrutinize, for example, just like a few hundreds components. You see that some are kind of kind of enriched, while others are kind of de-enriched. And that's of course pretty cool because, and we have those at a very large concentration because then they're full. We get a full dense phase of these enriched kind of components. And I'm not saying that this solves a conundrum, that's very important. It just could be one part because you just roughly get it for free, cycles and phase separation in these kind of systems. Okay, this is it for today. I'm sorry for being a little over time. I showed you a little bit kind of, at least I just showed you the principles. I mean, if you're really interested, we can chat much more how we deal with these very high dimensional mixtures, etc. I showed you that it works, this kind of selection. And of course, I have to thank the people, my group. I mean, these biochemical reaction stuff is done by Sudarshana and Giacomo, and also Yashu was an internship with us. So working on this selection project. And big thanks, of course, to our collaborators because they are the greatest they give such great inputs for theoretical questions that are exciting to look at for living systems. And of course, it's also great to test our theoretical predictions to actual observations in living cells and kind of virtual life kind of context. And big thanks, of course, to Frank Jülicher and the entire environment here. Okay, that's it. Thanks. Sorry for being over time.
No, thank you so much. It's difficult because everyone's on mute, so people don't usually like clap and sync, but it's implied. That was really great. So if anyone has any questions, fire away now. You can just unmute yourself and ask. If not, then I can set the ball rolling. So I was just going to ask about, when you were talking about the droplet splitting at the beginning, so like that 3D simulation of the dividing, and then you said that there was like a similarity or an analogy with like an electromagnetic interaction.
No, there's an analogy to electrostatics.
Electrostatic, sorry.
I can do this electrostatic analogy just like because I think that connects to what I think you maybe solve at the very moment in your lectures. I mean, these equations are notoriously hard to solve because these are kind of transient nonlinear diffusion equations with very high order gradients, but you can actually derive kind of a more reduced description of effectively like, they look more like diffusion equations again. And you can analyze this in the limit of fast diffusion and do a so-called quasi-static approximation. And then what you end up with is a Laplace equation. And then imagine now, Laplace equation you know from electrostatics. And imagine now, for example, you have a dielectric or whatever kind of stuff, but not of a sphere, of a kind of a, whatever, a beam, okay? And you would calculate the corresponding electric potential that would then correspond exactly to the concentration field. You would see that at the large curvature edges of this beam, they are more densified. And more densified means, in the language of concentrations, a larger influx. And that's why there's a larger influx at the high curvature edges. And you've seen this, I think most of you have calculated this in electrostatics, typical kind of boundary value problems. So you can reduce it in some limit to a boundary value problem, and then you see exactly this densification very beautifully. And then this explains at least this larger influx, but still you need the fact that you degrade inside, and as you have this larger influx, then you create this knack because you want to conserve roughly the volume. That's the other extra bit. I mean, it's of course you always have to be careful. It's a very complex things that are going on in this movie, and we always want to make simple explanations, and but that's the way we see it at the very moment.
Ooh, very cool. So anybody else have questions? Come on, people. I know it's the end of term for us, so everyone's very tired. But anybody? Otherwise, I'll just keep asking. All right, I'll just keep asking. So we always ask, like, especially with biophysics, like coming from a physics background, like most of the people here, like how do you what would you recommend to sort of like get into that like field, or like if you're interested in it, kind of like reading or just like fun stuff to do to like find out more?
I mean, about reading, I would definitely recommend to read kind of review articles. And then also to cherry pick a little bit, kind of, is it written by a biologist or by a physicist, and to have these different viewpoints. I think that's very important because the field is very interdisciplinary. And some people might be afraid of it. I completely understand this because somehow it feels as a physicist, you have to understand how a microscope works, you have to think about experimental techniques, you have to know what is an error bar, all the things that theorists often try to avoid in their life, just sample data in the simulation, so I don't need error bars. And this is not possible in that field because you have to learn to talk to mathematicians, to engineers, to biologists, to experimental physicists. But I can promise you, once you give it a try, this is super exciting because because there's no universal understanding of this super complicated system. These are complex systems where we just try to improve our understanding. But the different perspectives from people of different fields, in particular from engineers, mathematicians, or biologists, it's really inspiring. So they explain the same stuff. So you work together, but with different words, but you realize then, okay, you benefit a lot. So you kind of complement your viewpoint, how in the end the biophysics of living objects actually work, and that's really inspiring. So I can highly recommend to jump over that barrier. Yes, it requires also I had to jump over this barrier, to be honest. I mean, and I didn't like it at first. I mean, I loved playing with equations when I was a student, and it's like, no, I'll never do anything different. So purely academic kind of interest. But at some point I realized there's a danger that you end up with toy models. So kind of to really try to scrutinize your beautiful theory with real life, just do it. It's worth it. It goes more messy. So once to get this theory curve on top of the true data, this can be really stressful, but it's really worth it because then you know, wow, the model I did can make predictions of something that exists out in this world. I'm not saying that all toy models don't do this, but I guess there's a huge collection of toy models that have a huge problem in terms of being practically irrelevant. And then of course we lose the beauty of making predictions for real systems. That's why. Give it a shot. It's really cool. It's really fun to talk to different people from different disciplines.
Yeah, shout out to.
Can I ask a question? Is that possible?
Yeah.
To ask a question. My name is Bob. I'm more from the biological side. I just wondered how stable the droplets are inside the cells, the sort of things that you were pointing out as structures in the nucleus and these sort of things. Are they there for the duration of the cell, or are they transient chemical factories coming and going?
that depends on the type of compartments you're talking about. There's some that just form upon stress and dissolve once the stress that's applied is gone. So it can be just seconds. Yeah, but because the formation all relies on diffusion, and if typical diffusion constants are like 1 meter squared per second, the cell is a micron big. 1 micrometer squared per second, and the cell is a few microns big. And thus, usually the build-up of these compartments goes within seconds or even milliseconds. So that's very quick. And thus, so I think it's a great strategy maybe for quick response. On longer timescales, when you think about even like successive cycles of divisions, I mean, I don't know a single example where the droplets have survived the division. They have reformed after division, but then typically the daughter cell is different and they have different properties, different sizes, different composition. That gets also very exciting. But usually then they dissolve really as it divides, and then the entire biochemistry turns mad, right? As the cell divides, and then usually because they're drops. I mean, drops are easy to form, but and also easy to solve, right? You just change a bit the concentration outside, so they can be rather transient structures. But I think that's not just their downside; it's also a beautiful property that you can just switch them off like a light very quickly, use them, dissolve them again, and then they're gone. It's not like building up a membrane-bound organelle. I mean, imagine you have to dissolve all the mitochondria all the time. So it's very costly, right? And that's why, in general, yes, but you have to look from case to case. At least on a cell cycle duration, there are some, like the P granules, that form very early, for example, in the C. elegans embryo, and they live until the cell divides. So the full cell cycle life of like 20 minutes, they are present, more or less. Yeah.
Awesome. Yeah, sorry.
So it seemed like mostly throughout your talk you spoke about DNA and proteins, and there are a couple of other types of biological molecules, and especially lipids. We think about forming membranes. So does this have any relevance to other types of biological molecules, or is it primarily only applicable to DNA and protein?
I mean, lipids, of course, have a larger variety of like phase transitions. They cannot just form oil drops. So if they are long enough and have these kind of two heads, they can also find these create these bilayer structures. Of course, they're highly relevant because they build exactly the compartments. And of course, looking at drops in cells doesn't mean that the membrane-bound compartments are less relevant. They are super relevant. And that's why also the research is like, and also we have lots of research, new research projects where we look at the interplay among these membrane-bound and membrane-less organelles. So we see them, and that's both in the physics of wetting of surfaces. So in the end, a protein drop full of DNA now wets in the end protein-rich lipid membrane. That usually has like little channels, and then that's exactly what it's doing. It doesn't typically homogeneously vat it, so it vats it at a certain position, and then locally maybe does something. That's what we would like to understand. Maybe transmits a molecule out of this kind of bag of molecules. Saying it differently, oil drops in cells, I've not seen that. So I think they're mostly used to build a more sophisticated structure like membranes. But as I said, we look at the interplay at the very moment, and that's why it's very relevant. I have to say one thing. That's why maybe explains the hidden question you're implying. The stuff is not phase separating because the molecules are somehow hydrophobic and the surroundings water-like and hydrophilic. That is the basic idea why, for example, an oil drop forms in water. This is definitely not the case in the living cells. So you're not phase separating from water. So actually the water density, the inverted protein density of inside and outside of a drop often differs only very weakly. What differs is the composition. So actually the phase separation, when I talk about phase separation, it's really certain proteins come together while others stay outside. But if we would look at the water content inside and outside, it can be different, right? It can be different, don't get me wrong. And generally it's very weak. So it's not a phase separation with respect to water. And that's really the big difference to a classical oil-water phase separation. And you also see this in terms of the concentration inside versus outside. So oil-water is typically 10,000 to 1. So it's a huge, like a strong difference. So you hardly find a water molecule in an oil drop. But for these protein drops, typically partitioning factors, as in living systems, I mean, like the P granules, if you look to a specific protein, and they're full of 20, 30 proteins, these P granules, for example, in C. elegans, they have like enrichments of 5, 10, because it's a multi-component system. So it's not like oil-water phase separation. It's not that highly concentrated, but still sufficient to make a huge difference. Yeah.
Do you know if like not lipids, but like sugars, like glyco molecules phase separate or something, because they can be pretty large, like polymers, and like I just don't know.
I mean, I think people know that in the field of classical chemistry. I mean, of course, it depends on the tail, right? I mean, they carry a charge. Charge in water usually makes it dissolve very nicely. It depends strongly on the tail. I think, to be honest, the field of phase separation is now looking at the contact to the membrane-bound organelles, but some people have always thought about how sugars are affected. But I think that's a rather new idea. So it's interesting that you ask it. So I can't tell much about it. Maybe, and because I'm not a biologist, I mean, I have to say that. Maybe some of my colleagues know more about it. Maybe there's something ongoing already. It's hard to keep overviewing the field, to be honest. I mean, otherwise I just read papers and stop science. So, but I have to confess, I have to look how many people have started thinking about it. I just saw one talk recently where people alluded to this idea. So I can't say much about it, what's the interplay to sugars. But in general, it's likely. So I would maybe look at a organelle that is associated to the metabolism. So potentially there should be maybe some sugars related to it. But sorry, I can't say much more, otherwise it's too speculative.
Cool. If anyone else has any questions, I mean, it's a really interesting field too. I think Chris will back me up, but like any questions are welcome, right? Yeah. Like, you know, as in like, don't worry if you think your question's stupid, but yeah, if that's it, then we will say like a massive thank you for Christoph for coming and talking to us, because non-Oxford speakers, like really nice to have someone outside Oxford Uni, because usually when we're not doing stuff on Zoom, those are the people who are sort of in town. So it's been really great, and everybody have a great, this is the last one of term, so have a great holiday, and keep doing physics, and thanks again for the great talk. Thank you.
Thanks again. Thanks. Thanks for the invitation.